Welcome back to the deep dive. For so many of us, our first real dive into genetics starts in a classroom with Gregor Mendel.
Right, the pea plants.
Exactly, the pea plants. He gave us these foundational laws, dominance, segregation. It all seems so simple. If you cross a tall pea plant with a short one, the rules tell you exactly who wins.
And his work was brilliant. It was the 101, the perfect starting point for these simple traits that fall neatly into two boxes. We call them qualitative characters.
Like having attached earlobes or not. It's either or.
Precisely. But the second you step outside that pea garden, the genetic landscape just gets, it gets messy. It gets way more complex and, frankly, a lot more interesting.
Okay, let's unpack this then. The sources we've got for this deep dive, they really get into that real-world complexity, all the exceptions that sort of prove the rule. We're jumping into this surprising world of non-Mendelian inheritance. This is the stuff that governs everything from why certain diseases can skip generations to why your skin color isn't just light or dark.
Our mission today is really to pull out those key ideas that explain why most traits in the living world just don't fit into a neat little Punnett square.
Right.
We're looking at traits that blend, genes that operate by committee, and patterns where the physical location of a gene completely changes the rules of the game.
So let's start at that basic level, the interaction between two alleles for one gene. The classic Mendelian view is that one allele is the boss, the dominant one, and the other is totally silent.
The recessive one, yeah.
But what happens when that relationship isn't so?
Yeah.
So black and white.
Well, we find out pretty quickly that dominance is much more of a spectrum, and there are two key patterns here. The first one is called incomplete dominance.
Yeah.
This is where the heterozygous, so an individual carrying one of each allele, ends up with a phenotype that's truly intermediate. It's a blend.
The classic example is always the flowers, right? Like snapdragons or Japanese 4 o'clock.
It's the perfect example. You cross a pure red flower with a pure white one, and the next generation doesn't come out red, it comes out
pink. It looks just like you mixed red and white paint.
Exactly. The product from that one dominant allele just isn't enough to make the full, rich red color. So you get, half the dose, and the result is a diluted pink color. It's a perfect compromise.
So if incomplete dominance is like mixing paint to get pink, what about the second pattern you mentioned, codominance? How is that different from blending?
Codominance is, it's a whole different thing. Here, both alleles are fully and separately expressed at the same time.
So they're not blending into something new?
Not at all. Both products show up side by side. Neither one masks the other. Think less about mixing paint and more about putting two different patterns right next to each other on a canvas.
Okay, that makes sense.
The textbook example for this is the human ABO blood group. If you inherit the allele for A antigens and the allele for B antigens...
You have tight AB blood, so your cells aren't making some kind of half A, half B thing.
No, not at all. Your red blood cells are literally producing both the A-type markers and the B-type markers. They are fully co-expressed.
And we can see this in animals too, right? Like with roan horses.
Yes. Or erminette chickens. The animal doesn't look gray or light brown. It looks speckled. It has distinct patches of white hairs and colored hairs. It's visible co-expression.
Okay, so we've covered the fluid relationships between two alleles. Now let's kind of zoom out. The ABO system is a great transition because while one person only has two alleles, the population as a whole has three options: AIA, IBOA, and AI dualers.
That's right, and that's a phenomenon we call multiple alleles. It just means that for one gene, there are more than two possible versions floating around in the population.
But an individual can still only carry two of them.
Still only two. But what about traits that are determined not just by multiple versions of one gene, but by many completely different genes all working together?
Now, this is where it gets really interesting.
This is the leap to polygenic inheritance, and this is what governs what we call quantitative traits.
Things like human height, or weight, or skin color.
Exactly. Things that don't fall into neat boxes. They show continuous variation. If you graph it for a population, you get that classic bell curve.
So it's not like you either have a widow's peak or you don't. With these traits, multiple gene pairs are all contributing a little bit. Their effects are cumulative or additive.
Precisely. For something like skin color, you've got several different genes, and each one contributes a small additive amount of pigment. The more pigment-adding alleles you have, the darker your skin tone. The fewer you have, the lighter.
And the sources bring up this incredible experiment by Nilsson et al. On wheat kernel color. It really proved this additive effect.
It's the perfect demonstration. So he crossed a dark red wheat kernel, let's call it AABB, with a white one, AAB.
The first generation came out intermediate, predictable.
Right. But then, when those F1 plants bred, the next generation, the F2, it didn't follow the old 9.3.3.1 ratio at all.
Instead, he got 5 different shades, 5 classes from dark red all the way to white, in this very distinct 1.4.6.4.1 ratio.
And that ratio is the smoking gun. It shows the additive effects. The big group in the middle, the 6, had a medium number of red-adding alleles, while the ones on the ends had either all of them or none of them. It was mathematical proof that 2 gene pairs were working together.
Okay, but genetics isn't just about color and height. Sometimes the proteins a gene codes for are so vital that a broken version can be, well, catastrophic. Let's talk about lethal alleles.
Yeah, this is where certain gene combinations are just not compatible with life. Every organism relies on thousands of genes for basic survival. A mutation can create a non-functional allele.
And often that's hidden, as long as there's a good working copy of the gene.
Exactly. But when you get 2 bad copies, things go south. And the most common pattern we see here is the recessive lethal pattern.
So the allele is only deadly in the homozygous recessive form.
Correct. The heterozygotes are usually fine, or they might show some, mild non-lethal change. The curly allele in Drosophila fruit flies is a great example.
The flies with 1 copy just have curly wings, but they live.
Right. But if you get 2 copies of the curly allele, you're homozygous, and you don't survive development.
And this is where the math gets weird. The Mendelian ratios we expect, like 1.2.1, they just disappear.
That's the key takeaway. If you cross 2 heterozygotes, you expect a 1.2.1 genotypic ratio, but that 1/4 of the offspring that gets the lethal homozygous combination, they die.
In utero or just after birth.
Sometime before you can count them, so that group is just removed from the data set.
Which means the ratio you actually see isn't 1.2.1, it's 2.1. You're just counting the survivors.
Exactly. It's a statistical artifact. You're looking at a population where 1/4 of them never even made it to the starting line.
Wow. Now, the really terrifying one is the dominant lethal pattern.
Yep.
Just 1 copy of the allele is fatal.
These are incredibly rare for a pretty obvious reason. If a single copy kills you,
You can't pass it on. The allele should just vanish.
it should, unless there's a delay. That's the crucial exception. The allele only sticks around if the lethal phenotype, the disease, shows up after you've already had children.
And the most tragic and well-known example here is Huntington's disease.
A truly devastating neurodegenerative disorder. It's caused by a dominant allele. If you are heterozygous, HH, you will develop the disease.
And the key is that symptoms often don't start until a person is in their 40s or even older.
Right. Meaning they've had decades to potentially and unknowingly pass that allele to half of their children. The source material notes this is also a trinucleotide repeat disorder, where the length of a repeated DNA sequence actually correlates with how severe the disease is. It's a genetic time bomb with a very long fuse.
So up to now, we've been focused on how alleles interact. Let's shift gears and ask where the genes are located, because that placement totally changes the rules of the game. Let's start with sex-linked inheritance.
Okay, so this is about genes located on the sex chromosomes. For humans, that's primarily the X chromosome. And because males and females have different combinations, XX for females, XY for males, the inheritance patterns get asymmetrical.
The key is that males are XY.
Right. For any gene on the X chromosome, males are what we call homozygous. They only have 1 copy.
So they can't be a carrier for a recessive X-linked disorder. They either have the good copy and they're fine, or they have the bad copy and they're affected.
Exactly. And that's why conditions like red-green color blindness or hemophilia are so much more common in males.
It also gives us a very clear rule for pedigrees. An affected father can never pass an X-linked trait to his sons.
Because he gives his sons the Y chromosome, he only passes his X to his daughters. And this unequal setup leads to one of the coolest visual phenomena in all of biology: X inactivation.
Ah, the calico cat example.
The calico cat, it's the perfect illustration.
So explain how those patches of orange and black fur connect back to non-Mendelian genetics.
Well, female mammals have 2 X chromosomes, right? But having 2 active sets of all those genes would be a problem. It's a gene dosage issue. So very early in a female embryo's development, each cell randomly and permanently shuts down one of its 2 X chromosomes.
So in a cat that's heterozygous for fur color on the XA, one X has the orange allele and one has the black. Some patches of cells will shut off the orange X and express black fur.
And other patches will shut off the black X and express orange fur. The result is that beautiful mosaic patchwork. And it's why calico cats are almost always female. It's proof that where the gene is located really matters.
Okay, so if that's about location in the nucleus, let's talk about extranuclear inheritance. This shifts our focus completely outside the nucleus.
Right. We're talking about genes that are located in the organelles, specifically in the mitochondrial DNA and, in plants, the chloroplast DNA.
These little power plants in our cells have their own DNA.
They do, and they follow their own rules. The defining rule here is absolute maternal inheritance.
Why is that?
It's all about fertilization. The egg contributes basically all of the cytoplasm and its contents, including all the mitochondria, to the new zygote. The sperm contributes its nucleus, and that's about it. Almost no cytoplasm.
Meaning you inherit all of your mitochondrial DNA directly from your mother.
Every bit of it. So if she has a mitochondrial disorder, all of her children, sons and daughters, will inherit those affected mitochondria.
But only the daughters can pass it on.
Exactly. That's the key distinction. Only the daughters pass it to the next generation. We see it in plants, too, like with variegated leaves in 4-o'clock plants. That's all controlled by maternally inherited chloroplasts.
Okay, for our final section, we have the most complex layer we pulled from the sources. This is about genes that are physically there, but are silenced based purely on which parent they came from. This is genomic imprinting.
Genomic imprinting it's a wild example of epigenetics. These are modifications that happen above or on top of the DNA sequence itself.
So the gene is there, it's spelled correctly, but it's just turned off.
It's turned off. You inherit 2 alleles for these genes, like normal, but for a small number of them, one copy is chemically silenced or stamped through a process called DNA methylation. This means only the allele from the other parent is actually expressed.
So it's like you have 2 instruction manuals, but the cell has put a big sticky note on one of them that says, "Do not read."
That's a perfect analogy. And what's really fascinating is how this gets reset every generation. The epigenetic stamps are established in the sperm or the eggs. They last for the whole life of the organism, but then they get completely erased and reset in the germ cells of the next generation.
So a man will erase the maternal imprints he got and put a paternal stamp on all his sperm.
And a woman does the opposite for her eggs. It's this incredible cycle. And it's non-Mendelian because just having the allele isn't enough. Its parental history matters.
And this has some really serious clinical relevance. If you only use one copy of a gene, say the one from your father, and you lose that copy, you have no backup. The maternal copy was already silenced.
Right. And we see the dramatic result of this on human chromosome 15. If the active paternal copy is lost, the result is Prader-Willi syndrome,
Characterized by things like chronic hunger and developmental delays.
But if the active maternal copy on that exact same stretch of DNA is lost, you get a completely different condition. Angelman syndrome, which involves severe intellectual disability and unique behavioral traits. Same piece of DNA, totally different outcomes, all depending on which parent's copy was lost.
We have come a very long way from Mendel's simple binary rules today. We've covered genetic interactions that blend, like incomplete dominance, and ones that express side by side, like codominance.
We looked at the committee approach of polygenic inheritance, where many genes add up their effects.
And we dug into the logistics of location, how being on a sex chromosome changes the rules, and how genes outside the nucleus are passed down only from the mother.
And finally, we saw how even information outside the DNA sequence, that epigenetic stamp of genomic imprinting, can decide which genes we actually get to use.
This deep dive really gives you the tools you need to understand the true complexity behind human traits and diseases. Whether you're trying to read a family pedigree or just understand the incredible spectrum of human diversity, you need these patterns.
And as a final thought to leave you with, we focus a lot on DNA in the nucleus and organelles. But our sources also mention this fascinating idea of infectious heredity.
Go on.
Think about it. Symbiotic or even parasitic microorganisms, bacteria, viruses that live in the cytoplasm can be transmitted exclusively through the mother's egg. This non-genetic invader can then be passed down through generations and have a huge impact on the offspring's phenotype. It really forces you to ask, what else are we inheriting besides just the DNA we normally look at?